Good morning, all. I, Ms. Shweta Patel, welcome you all for the one-day RUSA-sponsored international webinar Biofacet, organized by Department of Microbiology and Biotechnology of Patkar-Varde College. A warm welcome to our guest speaker, Dr. Evelyn Sattlegger, Principal, Dr. Teresa Joseph, Chief Coordinator and Coordinator of Microbiology and Biotechnology Department, Ms. Ruchita Dalvi, staff members, and all the participants. I would like to introduce you all to our beloved Honorable In-Charge Principal, Professor Dr. Teresa Joseph. Dr. Teresa Joseph holds MPhil and PhD in botany. Ma'am has received the Young Scientist Award from NESA, that is National Environmental Science Academy, in 1989. She has successfully guided PhD students and was topper at her BSc and MSc. She has published more than 40 research papers in national and international journals and is a life member of many reputed journals. She has worked as joint coordinator and subject expert in the tree census program of Brihan, Mumbai in 1999. I now request Professor Dr. Teresa Joseph to speak a few words and declare the webinar open.
Good morning, everybody. On behalf of Chikitsak Samuha's Patkar-Varde College, I welcome you all to this RUSA-sponsored international webinar, Biofacets, Protein-Protein Interaction Using Yeast to Hybrid Systems. Chikitsak Samuha's Patkar-Varde College is a prime institution in the western suburbs of Mumbai. Our institution was re-accredited by NAC in the third cycle with an A+ grade and an institutional score of 3.53, and autonomy was conferred on our college from the academic year 2019-20. We at Patkar-Varde College are constantly upgrading our courses and infrastructure to provide students with the best possible learning opportunities. In keeping in line with the vision and mission of our institution for the holistic development of our students, various departments organize workshops, webinars, conferences, etc. And today, the Department of Microbiology and Biotechnology of our college has jointly organized this international webinar. I welcome our international speaker, Dr. Evelyn Sattlegger from the University of New Zealand. We thank the management of Chikitsak Samuha, the President of Chikitsak Samuha, Shri Kishore Rangnekar, the Vice President, Dr. Kiran Mangaonkar, the Secretary of Chikitsak Samuha, Dr. G.V. Pandit, our CEO, Dr. Mala Karkar, for their constant support and encouragement always. I wish you all the very best for this international webinar. Thank you.
Thank you, ma'am. We at the Department of Microbiology and Biotechnology has been actively involved in organizing various seminars, conferences, workshops, and guest lectures for the students to gain insights on ongoing research in various fields of biology. Started in 2012-13, Biofacet is a step towards this initiative. We started with state-level seminars to national-level seminar and national level conference, and from the last 2 years, it has taken us international step amidst the pandemic, which did not allow us to step back. At international level, we had speakers from USA on topics of vaccine development at Pfizer and CRISPR-Cas. This year, the topic for Biofacet is protein-protein interaction, yeast to hybrid system. Let us now introduce to the speaker of the day, Dr. Evelyn Sattlegger. Associate Professor Evelyn Sattlegger studied biology in Hanover, Germany, a degree offered jointly by the Leibniz University, Medical School and Veterinary School. She obtained her MSc degree in microbiology genetics, biochemistry, and immunology. After obtaining her PhD, she conducted research as a postdoctoral fellow at the National Institutes of Health in the USA. In 2005, she moved to New Zealand to accept a teaching and research position at Massey University, Auckland campus. Currently, she is the group leader for the Molecular Bioscience faculty at the newly formed School of Natural Sciences. Since 2013, she is also an associate investigator with the Maurice Wilkins Centre, a New Zealand Centre for Research Excellence targeting serious health human diseases. And she has been a member of the Auckland Medical Research Foundation's Medical Committee for almost 7 years. Sattlegger's research group currently consists of 3 PhD students, 4 MSc students, and a research officer. Her group conducts basic and biomedical research aimed at understanding signal transduction pathways, function of proteins, how they influence each other. The group interests include molecular and biochemical mechanisms occurring in healthy organisms, how disease and disorders develop, and how these can be prevented or treated. Sattlegger's group applies a variety of techniques in the fields of molecular biology, biochemistry, genetics, cell biology to biotechnology. It is indeed our pleasure to have such an eminent personality amongst us. Before we begin with the webinar, I request all the participants to be kindly patient. If any technical problem arises during the webinar, we will try to resolve all the technical glitches. Kindly post your questions in the chat box. It will be answered at the end of the session. Please maintain the decorum of the webinar. We are sure that this webinar would certainly complement to your learning objectives. Now, I would request Dr. Evelyn ma'am to address the webinar. Thank you.
Thank you very much for the invitation. I feel honored to be able to talk to you today. At the moment, I cannot share my screen. Now it is working. All right. Can you all see the screen?
Yes, ma'am, it is visible.
Okay, perfect. Thank you. All right, so before I start talking to you, I would like to give an overview of what I'm going to talk to you today. So I will talk about proteins, protein-protein interaction, using Baker's or brewer's yeast as a model organism to study protein-protein interactions. Then I will go further into the details of scoring protein-protein interactions, the advantages of protein tags, and then I will just quickly summarize what I have talked about. Okay, so let's go to the first topic, which is about proteins execute the biological work in cells. So it is the proteins that execute the biological functions in the cell. So just some examples here, oxygen transport, when you think about the red blood cells, when it comes to structure, hair, skin, and bone, nerve signaling, enzymes, muscle contraction, making proteins themselves, DNA, the immune system, and also the metabolism. Okay, so again, proteins are the workhorses that execute the majority of biological functions. So the cells have to execute a lot of different biological functions. So that means they need to have a huge repertoire of many different proteins. Okay, and so if we just look at one protein now as an example, so the DNA has the information on how to make a protein. So there is a specific gene for each specific protein that contains the information on how to make that protein. So the DNA is being transcribed in order to make the mRNA, which is being translated at the ribosomes to produce the protein. In this process of translation, it is the tRNA, which is a specific form of RNA, which delivers the building blocks of the proteins, which are the amino acids. All right, so a cell contains all genes, so all possible proteins that can be produced. but the cell doesn't need all of these proteins at all time. Or when you think about tissues, tissue cells need a specific type of proteins, and other tissue cells need a different subset of proteins. So that means there has to be some type of regulation, because protein synthesis is expensive to the cell. So it only wants to make the proteins it actually needs. In addition to that, the cells are also exposed to different environments. So therefore, the cell needs to produce proteins that are necessary for this particular task or environment. So basically, there has to be some spatiotemporal regulation of what proteins are in the cell, that they execute the biological functions that are necessary at a particular point in time and in a particular tissue. So that means a cell adjusts to this changing environment by changing their repertoire of proteins. So this can be done by the regulation of gene expression. So there can be transcriptional regulation, and then there can be translational regulation. And so this basically determines what gene is being expressed and how much it is being expressed. So at the end, what protein is being produced and how much of this protein is being reduced. Then there is another level of regulation that is possible, and that is by altering protein function. So the proteins are already present in the cell, but now it is being determined whether the protein is actually dormant or whether it actually is functional and it's doing its job. And so this is called the post-translational regulation. And one example is the protein kinases that phosphorylate a specific protein, so attaching a phosphate group, in order to turn this protein on or to turn it off, depending on what type of protein it is. Another type, if you want, of post-translational regulation is actually protein interaction. And that is what we come to the next point of my talk, which is protein-protein interactions, which is very often abbreviated as PPI, as a critical that these protein-protein interactions are critical for proper cell function, because proper protein function means proper cell function at the end. All right, so very simplified, again, proteins, the important thing is proteins do not work alone. Their activity is often regulated by other proteins binding to them. So in this example here, you have an enzyme, and then you have regulatory proteins binding to this enzyme through protein-protein interaction. And then you can have another regular protein that binds to that regular protein through protein-protein interaction to control how the yellow regulatory protein is interacting with the enzyme. So you have a regulatory cascade. On top of that, you can have other regulatory proteins that, independently of the yellow regulatory protein, can bind to your enzyme and regulate it again. So that means you can have a whole network of protein-protein interactions to exactly fine-tune the activity of the enzyme, that it has the appropriate activity for the cell that it needs in this particular point in time. So again, this shows that protein-protein interactions are essential for proper cell function. So just to show an example, when it comes to the research we are doing, we are interested in a protein called GCN2, which is a protein kinase that phosphorylates EIF2 alpha. And so this GCN2 protein kinase is in charge of many different functions in the cell. So you can imagine it needs to be very tightly regulated, that not something, something goes totally wrong. So for that reason, studies have already shown that there's a huge regulatory network of protein-protein interactions, which are indicated here in black lines. So all these circular green ones are proteins that activate GCN2, and the stop sign in red are the ones that inhibit GCN2. And so these interactions don't occur all at the same time. So it's regulated at specific times and specific tissues. So there is a dynamic happening. So again, you can see it's important to understand these protein-protein interactions to understand how GCN2 is functioning properly in the cell. And there are actually more proteins that control GCN2, where we still have to find out how these protein-protein interactions work in this particular case. So you can see in this example how complex it can be. All right, so coming back to the simplified image again, so you have these protein-protein interactions, which are important to determine the exact activity of enzyme that is necessary for the cell in that particular time. So you can imagine if, due to a mutation, for example, a protein-protein interaction is disrupted, that will have severe impacts on the cell. So it will impact the activity of the enzyme and may lead to diseases and maybe even to cell death, because this enzyme is not properly regulated anymore. Okay, coming back to protein-protein interactions. So you can see they are important for regulation. Sometimes they happen, sometimes they don't happen. And in this case, you can imagine it is very important that these interactions are weak so that you can dissociate and break these interactions when necessary, and then that the interactions can occur again. So they cannot be strong interactions. They have to be weak interactions so they can be dynamic. And so the three major interactions that can occur are ionic interactions, because opposite charges attract, hydrogen bonds, and hydrophobic interactions. Okay, so this is in the cell how it can be dynamic, because these are weak interactions. But at the same time, that means if you want to study protein-protein interactions, you have to keep in mind that they are easily disturbed by wrong salt concentrations and by the wrong pH. So if you want to study protein-protein interactions using purified proteins in the test tube, it is absolutely important to make sure you have the correct salt concentrations and the correct pH. But of course, you can imagine circumventing this problem. If you study protein-protein interactions in the living cell, it is under physiological conditions where it already has the correct salt concentrations and the correct pH. So meaning studying protein-protein interactions in the living cell might show you more what is really happening in the living cell. All right, so just to reiterate, to understand how diseases develop and how you could treat them, and also in order to make organisms that work for you, like biotechnology, and you want to engineer them in a way that they do jobs for you, we first need to understand protein functions at the molecular level in detail, which includes the protein-protein interactions. And as I have already indicated, the most straightforward thing is to study these protein-protein interaction in the living cell. Well, studying on humans and mammalian experimental animals is a little bit difficult to do. So instead, you can use mammalian cell lines and study protein-protein interactions there. But again, when it comes to mammalian cell lines, cell culture work is laborious, expensive. The doubling time is 24 hours, maybe even longer. Some of these cells, like primary cell lines, don't live very long. So alternatively, you could think about, I could use Escherichia coli, which is well established in the laboratory to work. It has a doubling time of only 20 minutes. It grows very quickly. But the problem is it's a prokaryote. It's a bacterium. It's not the same as mammalian cell lines. There are huge differences. So that's a disadvantage. So that brings us to Baker's yeast, which is my third topic, Baker's brewer's yeast as model system for molecular studies. So Baker's or brewer's yeast is exactly the organism you use to make bread or make beer, and the Latin name is Saccharomyces cerevisiae. Baker's yeast is a eukaryote just like mammalian cells. It has a nucleus, vacuole, all the organelles that mammalian cells have as well. So are very, very similar. And at the cellular and molecular level, yeast and mammalian cells are very similar. Yes, yeast is not a neuron, but at the molecular level, when you look at transcription, when you look at proteins interacting with each other, at these molecular basis of functioning of cells, metabolism, regulation, signaling, it's very, very similar. And in fact, in 2001, the Nobel Prize was given for finding the first genes responsible of cancer using yeast as model organism. And other examples, yeast can be used actually really as a model for neurons. It can be used for drug development. It can be used to understand human diseases. And it's actually used also for making vaccine, like the human papillomavirus. All right, so just another example when it comes to our research. I told you GCN2 is the enzyme we are interested in. And the function of GCN2 in yeast and mammals, it is the same when it comes to certain aspects. It does exactly the same whether it's yeast or mammals. In mammals, it has acquired some additional functions like long-term memory formation. But again, at the molecular level, it is actually very similar if you just look at the protein itself, what it is doing. And so we are using Baker's yeast as a model to understand the diseases associated with GCN2. So you don't have to read all of this in detail. But the examples of diseases, what we are working on is cancer and epilepsy. All right, so again, at the molecular cellular level, yeast and mammalian cells are very similar. So you can take yeast as a model organism to understand how mammalian systems are working. And yeast behaves almost like a bacterium. So it grows quickly. It can be handled very easily. Now I would like you to introduce you a little bit to Baker's or Brewer's yeast. So the yeast grows by budding. So you have here the yeast cell and you have the little bud emerging here, and then it separates from the mother cell. Yeast can exist as a diploid cell, just like human cells. But yeast can also exist as a haploid version, and you have the mating type A and the mating type alpha. And of course, these two haploids can mate. Basically, they just fuse together to then generate the diploid cell. And the diploid cell behaves exactly the same as the haploid cell when it comes to growing them and studying them. And then the diploid cell can undergo meiosis. So as a result, you have again haploid cells, 4 of them. Out of one diploid cell, you get 4 haploid cells, and they're enclosed in a sac, which is called a zygote. And then these haploid ones can grow out again into your haploid yeast cells over here. So when it comes to research, researchers prefer to use the haploid cells. The reason being that you only have one set of genes and not 2 sets of genes. So if you want to mutate something, mutate a gene, you don't have to worry about the other gene because you only have one set of genes. So that makes it very easy to work with when you look at, the genes and the effect on the cell itself. All right. So yeast, as I said, is very amenable to many molecular techniques. So I'm going to introduce you to some things that you can do to yeast. So here's again your yeast cell up here. And yeast can actually maintain plasmids, which is a circular DNA which exists in the cytoplasm instead of the nucleus. So yeast has a nucleus with its genetic material, but you can introduce plasmids into yeast just like you do with Escherichia coli in the lab. And so these plasmids look very similar like the ones used in the bacteria. So this is a plasmid you use in Escherichia coli in the bacterium. So you have here a multiple cloning site where you put in your gene of interest you would like to express. You have a replication or origin of replication so that the plasmid can be replicated and passed on to the daughter cells. And then you also have a marker gene. In this case, it confers resistance to the drug ampicillin so that you can maintain the plasmid. So you grow the bacteria on ampicillin, and then it must maintain the plasmid to survive. Okay, so now for yeast, the system is very similar. You use the plasmid, but in addition, it contains a set that is particularly for yeast. So there is a origin of replication for yeast, and you have a selectable marker to maintain the plasmid in the yeast. And so this selective marker confers prototrophy to uracil. So uracil is part of your genetic material, which is essential to life. And the yeast cells you use lack one gene that is necessary to make this uracil. So that means if you put this plasmid in and you grow the yeast on medium lacking uracil, your yeast strains, only when they have the plasmid, are able to survive and grow. And that way you force the yeast to contain that plasmid. So that's how you maintain the plasmid in the cell. All right, So I have already introduced you to the zygotes, right? So these are these haploid cells after meiosis. So you have a set of 4. And so this is very, very nice to use to study inheritance and segregation of genes, and also to engineer yeast with specific gene, because you can do targeted mating. And so what you do is you take these zygotes and use a micromanipulation microscope to take these individual cells out of each individual zygote and line them up on an agar plate, those 4, and you grow them, and then you investigate them, what properties they have. And then you can see how genes segregate. And so we have one of these ones in our lab to do these separations. Another thing which is very easy to do is also gene engineering. So let's just imagine you have this gene and you would like to knock that one out. And what you want to do, you want to replace this gene by your selectable marker, which is the uracil one here. And so by PCR, you can make this construct here. So you have this gene, and you have adjacent to it the flanking DNA that is homologous to the flanking DNA of your gene. And so it only has to be 50 bases, which is not much. If you did the same in mammalian cell lines, you need to have kilobases to make it work. And even then it doesn't work very well. But in yeast, with only 50 bases here of homology, it is very precise and very efficient. So you have homologous recombination here, and then your gene gets swapped out by the uracil gene, and now you have this one here. So your gene has been replaced by the uracil, and your yeast cell now is deleted off your gene of interest. And what you even can do as well is you can directly construct a plasmid in yeast without using restriction site, without using a ligase, so without using any enzymes or doing something in a test tube when it comes to making that together. So imagine you have this plasmid here with your gene of interest, and what you want to make is the plasmid with the same gene of interest, but with a mutation in here. So what you do is you cut out this area here by PCR, you make the same area, but containing the mutation here. And then you introduce both of these pieces into the yeast cell, and then it does recombination for you, and you generate a yeast cell that has the intact plasmid. And so this allows you to make really fast screening, because the resulting yeast strain can be used straight for investigating the effect of the mutation on that gene. And that brings me to one assay, which is later on also important for the yeast to hybrid system, which is the semi-quantitative growth assay. So you grow the yeast, for example, here in a glass tube, and then you transfer it to Eppendorf tubes and subject it to 10-fold serial dilutions. And then you transfer a small volume to a solid agar plate. So you see these little spots here. And then you can do that also with more of these ones, so that you have several of them next to each other on the plate. These plates, you put them to 30 degrees so that they can grow. And then you investigate how fast they can grow. And so you can see here, you have these serial dilutions. And so, for example, this strain here grew very well, but you can say that those two didn't grow very well. So that means the function of this protein was not really there. So by growth assay, you can deduce what the effect of the mutation was on that protein. And so here you can see one example. You don't have to look at all of this. Just look at these arrows here. So what we did in the screen was we looked at gain of function. So that means improved growth. And where you see the arrows, you can see that they grow better than these neighboring ones here and this control strain over here. So we did a series of point mutations, and then we tested them all on the plate to find out which point mutation led to a gain of growth. So you can see these two here grew very well. So there was a full gain of growth in those two examples here. All right. And then, of course, the other thing you can do in yeast is to score for protein-protein interactions in the living cell. And so the general principle for scoring protein-protein interactions in the living cell is when the protein-protein interaction occurs, this leads to a protein being expressed or being activated, which then leads to a scorable phenotype. Basically, it leads to something you can see. And that then helps you identify that the protein-protein interaction has occurred. And what you can see, what you can use, for example, as a mechanism to score that the protein has happened, is to look for gain of growth or loss of growth, or the production of color or the production of light. So totally dependent on the type of protein-protein interaction and the system you're using. These are the things how you can make it visible by the eye that the protein-protein interaction has occurred. So that brings me to the next topic, which is the scoring of protein-protein interactions. So first, before I do that, I would like to quickly introduce you to transcription, how it happens in eukaryotes. So in eukaryotes, you have a gene and there's a promoter in front of the gene. This is the RNA polymerase that transcribes the gene. But the RNA polymerase on its own is unable to find the gene that needs to be transcribed, and it's also unable to find out where it has to start transcribing. So for that, you need to have a transcriptional activator. And so in this example, it is the GAL4 transcription activator. It has two domains. One domain is in charge of DNA binding to the promoter, finding the promoter that is the one where the gene should be expressed. The other domain is the activation domain. The job of the activation domain is to recruit the RNA polymerase so that now the RNA polymerase starts transcribing this gene. So in other words, the RNA polymerase must have a transcription activator in order to be able to transcribe this gene. So now the yeast-2 hybrid system takes advantage of the fact that the DNA binding domain and the activation domain need to work together in order to recruit the RNA polymerase. So if you cut this protein in two and you express them separately, you will not have a transcription of this gene. The DNA binding domain is able to bind to the promoter, but because it's not physically linked to the activation domain, it is not able to recruit the activation domain, and therefore there is no transcription happening. So now you have your genes of interest you want to study for protein-protein interactions. Okay, so these are the two genes of interest, and let's just say we know they interact with each other. So what you now do is you fuse one protein to the activation domain, the other protein to the, one protein to the DNA binding domain, the other protein to the activation domain. So the DNA binding domain binds to the promoter and it's linked to your protein of interest. But because your two proteins are able to interact with each other, now these interact, thereby the activation domain is brought close to the promoter. It is able to recruit the RNA polymerase and transcription can occur. So in other words, only if your proteins interact, this gene is going to be expressed. All right, so here we have it again. Only if your proteins of interest interact, this gene is going to be expressed. Okay, great. But how can you score whether this transcription is actually happening? To do that, what you do is you link a so-called reporter gene to this promoter. And so the reporter gene, there are different reporter gene. You link a reporter gene to the promoter. And there are different types of reporter genes you can use. And I have here 3 examples. Okay, again, reporter genes are used to score for the rate of transcription. So you could have the LAC-Z gene, so the gene coding for LAC-Z, which makes the beta-galactosidase. So that means you have a colorimetric assay. So that means the yeast cell will turn blue if there is a protein-protein interaction. If there's no protein-protein interaction, then no color will be generated. The other 2 reporter genes, for example, is the His3 gene or the ADE2 gene. So the His3 gene is important for making histidine, which is essential for the cell to grow. Adenine is important for, the ADE2 gene is important for making adenine, which is again important for the yeast cells to grow. So you use a yeast cell that is lacking these genes, And instead, it has the His3 and the other two gene linked to the promoter you're using for your yeast to hybrid assay. So that means only if there is a protein-protein interaction, those cells can grow, because only then they make histidine and adenine, and they're able to make their own histidine and adenine and grow on the medium. If there is no protein-protein interaction, they don't make histidine, and therefore they are unable to grow. So that means with LAC-Z, you look for color, which tells you there's an interaction happening. For His3 and other two, you look for growth, whether cells can grow or not, as an indicator for protein-protein interaction. So let me show you one example. So here we have GCN1 and RPS10, and this is to score whether those two interact with each other. The reporter genes are His3 and other2, so that means we're using a strain that is lacking the genes His3 and other2 in their genetic material, and we bring these two reporter genes into the cell. So the interaction is happening. Therefore, these two genes can be transcribed, which means they can make their own histidine and adenine, and therefore they are able to grow on medium lacking histidine because they make their own histidine. They're able to grow on medium lacking adenine because they can make their own adenine, and they can also grow on medium lacking both histidine and adenine because they have both of these reporter genes. And so as a control, for example, if you have RPS10 bound to the activation domain, but the binding domain is not bound to the other protein, then of course you don't have any interaction happening and your genes are not turned on. And that's what you can see here in this control. There is no growth because these genes are not expressed and therefore the yeast cells cannot grow. So again, in this system, yeast to hybrid, you look for cell growth as an indicator for protein-protein interaction. All right, so this is just another image saying the same thing. So here you have your protein-protein interaction happening in yeast to hybrid. So here is the DNA binding domain, here's the activation domain. If this interaction is happening, your activation domain can be recruited close to the promoter and transcription can happen and you see cell growth. That's the yeast to, that's the classic yeast to hybrid system. And then, of course, there are other varieties of the yeast to hybrid system I'm going to introduce to you now. One of it is called the reverse yeast to hybrid system, which works exactly the same as the yeast to hybrid system. But in this case, you have a reporter gene that when it gets expressed, it causes toxicity and kills your yeast cells. So that means in this case, you're looking for lack of growth as an indicator for a protein-protein interaction. Then in another example, the yeast 1 hybrid system is, for example, a study of, that's where you can study a DNA binding protein. So here your protein of interest binds to the promoter, and then you have the activation domain linked to your protein of interest, which recruits the RNA polymerase. So here you can investigate whether a mutation abolishes your protein to bind to the promoter, because then the reporter gene is not being turned on. So here's another example, which is now called the yeast 3 hybrid system, which basically means that 3 components are involved in mediating an interaction. So you have 2 proteins of interest, and then in the middle here, you have a molecule that is necessary to actually mediate the interaction between those 2 proteins shown here in black. So that means only if this mediator molecule is able to mediate this interaction, then your activation domain is recruited and transcription can occur. So that's why it is called a yeast 3 hybrid system. And this mediating molecule could be another protein, could be RNA, DNA, a drug molecule, anything, basically. All right, so now the yeast 1 hybrid, 2 hybrid, and 3 hybrid system, because it is based on transcription, it is based on recruitment of the RNA polymerase and transcription, it has to occur in the nucleus. So some of the protein-protein interaction might not be able to do in the nucleus, because in order to do the yeast 1, 2, or 3 hybrid system, your proteins of interest need to be transferred into the nucleus. And so sometimes with these studies, it actually might not be possible to get these proteins into the nucleus. And so there are other hybrid systems you can use instead that occur in the cytosol instead of the nucleus. And so here's one example, which is the membrane-based yeast 2 hybrid system, or also called the split ubiquitin assay. So this circular thing here and that circular thing here is the ubiquitin, and it's split in half, so N and C terminus. And so it is linked to your proteins of interest, which happen to be membrane-bound proteins of interest to investigate whether they interact with each other or not. And if these two interact with each other, they bring your ubiquitins, those two halves, close together so that they can reconstitute. And that allows now the cleavage of a transcription factor of your ubiquitin, which then travels into the nucleus to recruit your RNA polymerase and turn on transcription. Okay, so you have a traveling only of a transcription factor into the nucleus, but your protein of interest is located outside the nucleus. In this case, it's actually a system to look for membrane proteins interacting with each other. There's another system, for example, this one here. So this is an enzyme complementation assay. So in this case, it's an enzyme that is also split in half. And so it is again linked to your 2 proteins of interest. And if those 2 proteins of interest interact with each other, that brings those halves of the enzyme close together so that it reconstitutes back again to a functional enzyme, which then can catalyze a reaction from a substrate into a product, which, for example, has a color, and then you can see that this is being produced as an indicator that protein-protein interaction has occurred. As one example of enzyme you can use is the luciferase. Okay, so in this case, you have here the luciferase that was split, and when your proteins interact, the luciferase is being reconstituted into a functional enzyme, which then uses luciferin and turns it into a light signal, which you then can see. All right, and now I'm going to introduce you to another system which also produces light, but not by an enzyme, but by a protein that is fluorescent, and that is your yellow fluorescent protein, YFP. So this yellow fluorescent protein, again, is cut into 2 pieces, and your proteins of interest each are fused to the N and the N and the C terminus of your protein. So if your 2 proteins interact with each other, that brings those 2 halves of YFP close together, your yellow fluorescent protein, which then becomes fluorescent, and that's what you can see under the microscope. And so this is an assay where you can study protein-protein interactions in the cytosol, but you can actually do it in the nucleus as well. So that means you can actually, under the microscope, see where in the cell does the protein-protein interaction occur. The other neat thing with the system is you can actually follow the protein-protein interaction with this illumination and expose the yeast to different environmental conditions to find out when does the protein-protein interaction occur, under what conditions does it occur, because you have the living cell under the microscope and you can see when the interaction happens and when when the interaction doesn't occur, because then the luminescence is gone. All right, so here we have one example. So we have here YRH1 fused to the N terminus of the venous fluorescent protein, and CDC28 fused to the C terminus of the venous fluorescent protein. And so here you have just some other 2 proteins fused to the N and C terminal half of the venous protein, where we knew that they don't interact with each other. So that's just the control, basically. And so there is some DAPI staining where you stain the nucleus, that you can see where the nucleus is. And then you also look at the appropriate to look at the BIFC signal. And you can see here there is no signal as expected, but here you can see a signal. And so now you can fuse the signals of BIFC and DAPI staining together. And here you can see the nucleus illuminating in purple, and in yellow, and in green, the BIFC illuminating. So that you can see in the living cell, you can see here the interaction is happening in the cytosol. All right, so coming back to the classic yeast to hybrid system. So I have told you that the interaction is being investigated by the DNA binding domain fused to one protein and the activation domain fused to the other protein. And if these interactions occur between new proteins of interest, then the activation domain is recruited close to the promoter. And then the polymerase, the RNA polymerase, can be recruited and transcription is happening. And of course, in order to do this study, you have to introduce into yeast plasmids that code for your constructs, right? So here's a plasmid that codes for your DNA binding domain fused to your protein of interest. So that codes for this one here. And this is the plasmid that has the activation domain fused to your other protein of interest, which then expresses this one here. Okay, so you have to bring in these two plasmids in order to express those two protein constructs to study protein-protein interaction. All right, so that means you have to make yeast and you have to bring both of these plasmids into yeast. So imagine you want to study several proteins and see which one binds to which one. So you, for example, 2 different types of this one, 2 different types of that one. So you would have to do several yeast transformations. So the other possibility to get those 2 plasmids into yeast is by taking advantage of yeast can also occur as a diploid. So you can make your haploid type A transform into it all the proteins fused to the binding domain, and the other haploid yeast, all of them fused to the activation domain. And by mating, you produce a yeast cell that then have both plasmids. So that's another neat way to bring both plasmids into one yeast cell to then do your studies. All right, so now I have talked to you about basically studying protein-protein interaction. So imagine you know a protein that binds to another protein, but you don't know which one. And by the way, it doesn't necessarily have to be yeast protein. It can be plant proteins or human proteins. It doesn't matter. You can express anything in yeast. So to find out which protein binds to your protein of interest, so this is the one here, you want to find out what other protein binds to it. You have a plasmid with the DNA binding domain and your protein of interest fused to it. And then you have a library, which basically means a collection of plasmids where the activation domain is fused to each single protein you're interested in. There are libraries available where you have all human proteins fused to the activation domain one by one. So a huge library with 10,000 of proteins, all the human proteins. And so with this study, you can investigate this protein here, to what other proteins does it bind to? So what you do is you transform into these yeast cells all the one plasmid with all this protein of interest with the DNA binding domain. And then in each single yeast cell, you have the different varieties of the activation domain bound to all these different other proteins. So each yeast cell has a different protein fused to the activation domain. So now what you do is you do the reporter gene assay, right? Because you know that if there's no interaction, your reporter gene is not being expressed. Only if there is an interaction between your protein of interest and the other protein, then you have transcription. So you can do the growth assay then, and only the ones where there is an interaction, you should see improved growth. And that way, in this case here, you can see that 2 out of the 5, you have a protein-protein interaction happening. Okay, so imagine now you have 40,000 proteins you would like to investigate which one binds to your protein of interest. That would be a lot of growth assays. So instead, what you can do is you can do a competition study. So what you do is you have all these yeast cells, but instead of these growth assays on solid medium, you bring all the yeast into liquid medium, all of them, all 40,000 you have, and then you let them grow in the liquid medium, because what happens is the ones who don't have the reporter gene turned on are not able to grow. But the ones who can turn on the reporter gene are able to grow and they multiply. So that means you're selecting for the ones where you have protein-protein interaction. And then, because we have all this large-scale sequencing, possibilities of large-scale sequencing, and there are some other molecular tricks I don't have time to talk about, but you can easily find out which of the ones grew and what are they coding for. All right, so this is basically a library screening. Okay, so you find out which protein binds to your protein of interest. All right, then of course you can do another screen. Imagine you know which proteins interact with each other. You're interested in two proteins interacting with each other, but now you're interested in one of the proteins, which amino acids are mediating the protein-protein interaction. So what you do is you make a library, which is a collection of genes, of your gene of interest, and each plasmid has a different point mutation. Okay, so you basically do mutagenesis, and each one has a point mutation. And then you bring these plasmids again into yeast, where your protein with the DNA binding domain is, the plasmid with the DNA binding domain is the same for every yeast cell, but each yeast cell contains the other protein fused to the activation domain with a different mutation. Okay, so now you do the same screen as before, right? So in this example now, it is not about growth, but it's about color. So you have the LAC-Z reporter gene fused to it. So that means if you have a protein-protein interaction, the LAC-Z is being expressed and your colonies turn blue. If there's no protein-protein interaction, there is no color. Okay, so the blue color indicates protein-protein interaction has occurred. What you are interested in are the ones where it is not blue, because the protein-protein interaction has been disrupted, which means that this particular mutation has affected the interaction, meaning that this amino acid that was targeted by mutation is responsible or required for the protein-protein interaction. Okay, great. But if you think about it, there can also be a point mutation that introduces a premature stop codon and your protein is truncated, and you get a false result because it says there is no protein-protein interaction, but actually that is happening because your protein is truncated. And so there's a trick what you can do, which is you fuse to the end of your gene a part that expresses another domain that confers resistance against the drug G418. Okay, so you have this one fused to it, and now you can see your proteins have this extra domain over here, and that confers resistance to G418. And that's what you can see here on G418. There is growth when the domain is present because it confers resistance to G418. So that means here, the one you're interested in, where there is no protein-protein interaction, you have no blue color. This strain can still grow on G418 because the protein is full length, has this domain, and confers G418 resistance. But the ones where you have a premature stop codon and your protein is truncated, this domain is not present, and therefore they cannot grow on this medium. All right, so this is a neat way of making sure that you not accidentally find the ones where you just had a truncation rather than a point mutation. So these are just examples what you can all do with yeast when it comes to looking at protein-protein interactions. All right, so this was all about now the yeast to hybrid system. And as you can see, it is extremely powerful to look at protein-protein interaction. Also keep in mind, the stronger the interaction, the more the gene is being transcribed. So that means it's even quantitative. Okay, so in this example, the stronger the blue color, the stronger the interaction. The better the growth, the stronger the interaction. So that means it's even quantitative and gives you an indication of how strong an interaction is, not only that it is happening. But nevertheless, even though yeast to hybrid is extremely powerful, and we use it in our group and many people use it, at the end, it is indicative of a protein-protein interaction, but it actually doesn't show you per se that the interaction is happening. So therefore, people tend to also do some other assay to provide evidence that the interaction has occurred, which brings us to the validation of protein-protein interaction via, for example, co-precipitation assays. So you have here a yeast cell, you have elongated in pink the protein you're interested in, and the circular ones are binding proteins. And you want to provide evidence now that these binding proteins actually bind to your protein. So what you do is you break these yeast cells open, and then you bind this protein to, in this case, an antibody, which is covalently linked to a heavy bead that you can precipitate with a centrifuge. Okay, so you can really pull it down because the beads are being precipitated. And when your protein binds to the beads, it is being pulled down with these beads. And if your proteins, your binding proteins, are bound to your protein here, your pink elongated one, that means they are coming along with it. They are being co-precipitated with your protein. And therefore, it is called a co-precipitation assay. And then this one here, you can detect via Western blotting or mass spectrometry to identify your protein and the ones that bind to it. Again, because of the time, I have not time to talk more about that. So this assay I have just told you involves an antibody to bind it. And so in this case, this type of co-precipitation assay is also called a co-immunoprecipitation assay because you use an antibody. However, sometimes you don't have an antibody to do this experiment. So you need to find another way to do it if you don't have an antibody. And so that brings us to the fifth topic, which is protein tags. So protein tags are basically extra bases you add to the open reading frame of your gene. And so that adds an additional domain to your protein, the so-called epitope tag, which has a specific property which is advantageous for you when it comes to what you want to do. And there are epitope tags with different properties. So one, for example, is the so-called GST epitope tag. So in this example here, you have fused the GST epitope tag to your protein of interest. And then you have beads that are linked with glutathione. And your GST has an affinity for glutathione. So that means when you now break the cells open, your protein, the elongated pink one, will bind to the glutathione through your GST tag, which is fused to your protein. And that way you precipitate your protein. So you use the property of the epitope tag in order to precipitate your protein of interest. That is the trick of this tag. All right, so here we have an overview. I have just talked about the GST, which is an abbreviation for the glutathione S transferase. So this is actually a portion of an enzyme, and it's the portion that binds its substrate, which is glutathione. And that's the trick of the GST binding to glutathione. And there are many other tags people can use. For example, here, the second to last one, GFP, the green fluorescent protein. So this is from the jellyfish Acropora victoria. You can localize your protein in the cell because it glows. So you can see it under the microscope. In addition to that, there are also antibodies available to this GFP. So that way you can detect it as well, and you can do co-immunoprecipitation with that as well. Then there is the maltose binding protein, which is from the system in E. coli. And that one actually helps with protein folding. So if your protein has problems getting folded properly, you fuse this epitope tag to it to make sure that it's actually folded properly. Then there are some other tags like the MIC tag and the FLAG tag. And this is a series of amino acids, relatively short. This one here is synthetic. This one is taken from another human protein, a piece of it. And for either one of them, FLAG or MIC, there is an antibody available, which you can use in Westerns or in co-immunoprecipitation assays. The same also for the HA tag, which is a fragment of the surface glycoprotein of human influenza virus, where there's also an antibody available. And then finally, you have a hist tag, which is 6 histidines, one after the other. And the 6 histidines like to bind to bivalent ions. And you can use resins that have bivalent ions, for example, the nickel resin, and take advantage of the fact that your histidine tag likes to bind to these bivalent ions. And there are many other tags, so I'm just showing you some of the common tags that are being used. All right, so then at the end, I would like to give a summary. So fine-tuned protein function is critical for proper cell function, and protein-protein interactions determine protein activity. So there are lots of protein-protein interactions happening in the cell. And I have showed you that Baker's or Brewer's yeast is a perfect eukaryotic model system when it comes to studying protein-protein interactions and many other molecular and biochemical studies. Okay, so for in-depth molecular studies, such as protein-protein interactions. And I have shown you that epitope tags can equip proteins with additional properties to make your molecular studies easier. And so here, just an example with yeast, you can easily combine structural biology, yeast molecular biology, biochemistry, genetics. So in this example, we have here the YRH1 protein where we have now a structure and we can look at amino acids we are interested in, and then we can use protein-protein interaction studies here to investigate how point mutations affect this interaction. And that is followed by co-precipitation experiment to validate that the interaction has occurred or not. And so we have now a liquid handling robot to also expedite our growth assays. And so this is just my research group. I have PhD students and master students who do all the work. So our university has 3 campuses. So Wellington is the capital city of New Zealand. And so I'm located in Palmerston North, which is very close to Wellington. It's just a little bit of an hour drive to get to Wellington. And our school also has a campus on Auckland. So yeah, thanks very much for listening.
So I must say it's a wonderful webinar and very informative session, madam. So can we move ahead for our next part of the webinar? That is a question answer session.
Yes, sure.
Yeah, ma'am. So I would request all the participants, you can kindly post your questions in the chat box. So one by one, we can just try to solve those questions. Ma'am will answer that. Yes, ma'am. I have one question. You told that cancer has been first detected by using that yeast domain or the yeast gene. Can you just elaborate a bit on that?
Yeah, sure. If we think about cancer, cancer is about uncontrolled cell division. And yeast also has to divide. And the genes in charge of making sure that the cell division starts at the right time. So basically the cell is ready to do cell division. You know that the genetic material needs to be duplicated before the cell division can happen. And so these genes that code for the proteins that are in charge of duplicating the DNA, setting up everything that is necessary for segregating the genetic material in the daughter cell and into the mother cell, and then also to do the cell division, these genes are absolutely conserved between mammalian cells and yeast cells. And so what they have done is they have used the yeast cell to find out which genes are responsible for that. And so in that way, then they could deduce into mammalian cells when these genes are defective, that this affects cell growth and leads to uncontrolled growth. So they basically use the yeast system because it is so easy to work with it and so easy to mutate the genes. They use the yeast system to understand how cell division works.
Hello, ma'am. there are two more questions.
Yeah.
The first question is, compared to in silico approaches for determining PPI, what are the advantages and disadvantages of using yeast to hybrid system?
Okay. So in silico is extremely powerful to get an idea what amino acids might mediate the interaction. But at the end, we need to still check in the living organism whether these amino acids actually mediate the interaction. Because if you think of it, the in silico studies are based on biological findings we have made in the past. And so we don't know everything yet in biology. And so therefore, we still need to go, whatever we propose, we still need to go into the living cell to validate that this particular amino acid is required for protein-protein interaction. And so yeast to hybrid, for example, is one system to validate such interactions. In addition to that, sometimes the in silico method to look at protein-protein interaction, if you don't know enough how it works and what area of a protein mediates the interaction, it is not possible to propose in silico which amino acids mediate the interaction. So in this way, the only way is to go into the yeast system and do these individual mutations to find out what amino acids mediate the interaction. But the other neat thing is to kind of work together with in silico and within the living cell, because once you know the first amino acids that mediate the interaction, then you can start simulating the protein-protein interaction in silico to propose more amino acids that mediate the interaction, and then you can go back into yeast again and check basically whether this is the right prediction, and that's how it works. Not sure whether I answered the question.
Yes, ma'am. There is another question. What will happen if the low protein expression efficiency will be there in the yeast?
Oh, when there is a low efficiency in protein expression. Yes, that can happen. So one possibility is there are different promoters you can use. There's a whole collection of promoters which can be used. Some of them are expressed weakly and others are expressed much stronger. And one possibility is then to switch to a different promoter system to get a better expression. Another possibility is that the expression might be actually fine, but the protein is unstable and get degraded. And then a possibility is to add, for example, an epitope tag to increase the stability of the protein, that it doesn't get degraded as easily.
Ma'am, there is one more question. Could you please explain the protein truncation part in yeast to hybrid for screening of relevant amino acids in PPI?
Is this the question about removing constructs that have a truncation?
Yes, ma'am.
Okay. All right. In this example, so just imagine you don't know which amino acid mediates the protein-protein interaction. So you could do random mutagenesis, which means you have a collection of your gene, where in every gene, each of these genes, one amino acid has been mutated randomly. And then you do your studies to look at protein-protein interaction. But if you do the random mutagenesis, you might have changed an amino acid codon into a stop codon. So that means now your protein is truncated. You're doing these mutagenesis to look for loss of protein-protein interaction, because that means that the amino acid that has been mutated is actually necessary for protein-protein interaction. So you're looking for a loss of protein-protein interaction. But if accidentally there has been introduced a stop codon and your protein is truncated, of course there is no protein-protein interaction. And it's not because your amino acid is necessary for protein-protein interaction, but instead it's because your protein has been truncated. And so the trick is that at the end of the protein, you add a tag, an epitope tag that confers resistance to an antibioticum. So that means only if the protein is full length, it is able, the cell is able to grow. So that means if you have a truncated protein, then this domain is missing and the cell cannot grow. And that way you get rid of the ones that have truncated proteins. Does that make sense? Otherwise, I can bring up that slide again.
Ma'am,
Yes.
there is one more question. Now, this question is with respect to the advantages and disadvantages of using yeast to hybrid system. So the question continued is, if we have domain information of a protein, can we make them interact and study their effect?
Sorry, can you repeat it, please?
Yes, ma'am. So the question asked is, if we have a domain information of a protein, can we make them interact and study their effect?
So you mean if you have already some information on a domain, how can you use the yeast to hybrid system to get more insight?
Yes, ma'am. Yes.
Yeah, so what you can do is, for example, one interesting thing to investigate is to make sure whether the, the minimal region required for the interaction in that particular domain. So you could just use in your yeast to hybrid system just this little fragment of the domain and check whether the interaction is occurring in this region. So you basically find out the minimum region in the domain that is required for this interaction, making sure that you haven't missed on another area in this domain that might also be required for interaction. So you can basically double check whether your assumption or your findings are correct, that only this part of the domain is sufficient to mediate this interaction. So that's something, for example, you could do.
Yes, ma'am. Another question is, what are the chances of contamination by molds? And if yes, how can we avoid that contamination of mold? Why?
Okay. Yeah. So yeah, that is a good question. In bacteria, you use antibiotica, resistance to antibiotica, and that way you kind of kill everything else that wants to grow. But in yeast, you don't really use antibiotica in yeast, very rarely, but instead you look at prototrophy. But it's actually relatively easy to work. If you work close to a flame, you actually don't get that much contamination. Okay. It actually works really nicely.
Okay. So, ma'am, my next question is it possible to do co-transformation in the yeast to hybrid assay?
Yes, you can. Yes, you can. You can transform 2 plasmids at the same time, 3 plasmids at the same time. Yes, you can do that. Yeast is extremely efficient when it comes to putting DNA into it.
Okay. How this yeast to hybrid system plays an important role in drug discovery or drug and protein interaction? Does it play any important role? And what could be the mechanism if yes?
Yeah. So you could, if you imagine you have a drug that disrupts a protein-protein interaction, these are actually new directions when it comes to drug discovery, that people are now focusing on protein-protein interactions and thereby cure diseases or disorders. So you can use your protein-protein interaction and then expose yeast to drugs and see whether the protein-protein interaction is disrupted. And because, as I said, the reporter gene, the stronger the interaction, the stronger the reporter gene is being expressed. So you can also see how well the drug is able to disrupt the protein-protein interaction.
Correct.
So that's how you can do that. In particular, protein-protein interactions, you can do look at drugs that disrupt the interaction.
Okay. Thank you very much, ma'am, for patiently answering our...
there is one more question.
Okay,
sir, shall I go ahead?
Yes, sir, please.
Ma'am, the question asked is, what will happen if the protein-protein interaction is mediated by a strong interaction?
Right. Yes, you can still do a yeast to hybrid assay. You can still investigate how mutations affect this interaction, because if it is a strong interaction, there are, in this case, many amino acids involved in this interaction, but you can still investigate it.
Okay. If no further question, I would like to proceed ahead for the last valedictory part of our today's webinar. So we'll start with the vote of thanks. I deem it a great honor and privilege to propose the vote of thanks at the end of this wonderful international webinar on protein-protein interaction yeast to hybrids. First and foremost, I thank our guest speaker, Dr. Evelyn Sattlegger, Madam, for sparing the time from her busy to grace this webinar and sharing her knowledge with us. Thank you so much, ma'am, for this. Special thanks to all the participants for attending Biofacet and for their kind cooperation throughout the session. And I also want to express my heartfelt thanks to our management member, Dr. Gurunath Pandit, Secretary of Chikitsak Samuha, Dr. Marla Kharkar, Chief Education Officer, Mrs. Ruchita Dalvi, Chief Coordinator of Patkar-Varde College, and co-coordinator coordinators and coordinator of the departments of microbiology and biotechnology, who all are the driving forces of our college and encourages us to organize such webinars, conferences, workshops on regular basis. I want to extend my thanks to all the staff members for their efforts to organize this Biofacet. Now, just participant kindly note, the link for the feedback form will be shared in the chat box. It will be open and accessible for 30 minutes. Certificates will be sent on the mail within a time of a week. Thank you all once again and hope to meet you all again for more such initiatives undertaken by Patkar-Varde College. Thanks one and all. Thank you so much, ma'am, for your guidance, for making a time for us.
Thank you very much. I enjoyed it very much. Great questions.
Our pleasure to have this webinar, ma'am, and hearing so many things and clearing so many concepts from you all. Thank you, ma'am.
You're welcome.